Thank you to the organizers for inviting me for this talk. Today, I will be talking to you about oncogene additivity in the PI3 kinase pathway in cancer. My name is Neil Vasan, and I am an attending physician at Memorial Sloan Kettering Cancer Center. I am a physician scientist and a breast oncologist working on cell signaling in breast cancer. These are my disclosures. Oncogene activation is a hallmark of cancer, and there are multiple ways that oncogenes can be activated. Proto-oncogenes, that is, oncogenes that are not activated, can be modified in primarily three different ways: gene amplification, point mutation, or translocation. And the identification of these mechanisms and the resultant oncoproteins, that is, the proteins coded by these genes, has led to some of our blockbuster targeted therapies in cancer today. Now, PI3 kinase is an oncoprotein complex composed of two proteins. It has a storied history, first discovered in the 1980s as a factor bound to polyoma middle T, a viral protein, and later found to be a protein that phosphorylates or places a phosphate group on the 3' position of phosphoinositide, which is a lipid species. Now, at the time, this was quite novel because kinases up until then had been reported to phosphorylate proteins, and this was a lipid kinase, phosphorylating lipids. Later, it was found that the PI3 kinase gene product, or one of the PI3 kinase gene products, was similar to a product found in a retroviral oncogene derived from birds called ASC16. Going into more detail, PI3 kinase is a heterodimeric lipid kinase. It's composed of two different proteins that come together and have enzymatic activity. The genes that code for the PI3 kinase complex are two genes, PIK3CA and PIK3R1. PIK3CA, colored in in gray, codes for the protein P110 alpha, while PIK3R1, colored in green, codes for the protein P85 alpha. P110 alpha is the catalytic subunit. It's the business end of the molecule, if you will, while P85 alpha is the regulatory subunit. It's necessary for P110 activity, but in and of itself is not catalytic. And together, these proteins bind together and form the PI3 kinase alpha complex. The PI3 kinase alpha complex, as shown in the figure outlined in red, sits at the hub connecting in the cell, connecting extracellular signaling, mostly through receptor tyrosine kinases, with intracellular signaling, as shown in the diagram. PI3 kinase catalyzes the formation of PIP3, which is a lipid species with 3 different phosphorylations from the lipid species PIP2. Now, PIP3 is a potent second messenger molecule that sits at the cell membrane and recruits a variety of proteins to the cell membrane, most notably AKT. AKT is a protein kinase that leads to the phosphorylation of many downstream substrates that are important and necessary for a variety of cellular functions, including protein synthesis, metabolism, such as glycolysis, and cell survival. What makes PI3 kinase signaling rather complicated is that there are a number of genes coding for different catalytic subunits, different regulatory subunits, and also there is a cell specificity with many of these proteins. There are 4 different isoforms of PIK3CA, alpha, beta, gamma, and delta. There are also multiple isoforms and splice variants of PIK3R1, the majority of which are poorly understood. Adding to the complexity, the alpha and beta PI3 kinase complexes are expressed in all cell types, while gamma and delta are primarily restricted to immune cells, delta in lymphocytes primarily, and gamma in myelocytes primarily. And together, these different isoforms, splice variants, and cell types help diversify cell signaling through the PI3 kinase pathway. Now, PI3 kinase in the oncology space was elucidated in the 2000s by multiple papers, many from the group at Johns Hopkins. And these groups found in 2004 that single PIK3CA mutations are frequent events in human cancer, that they're sufficient for cell growth, and can also be inhibited by small molecules. And much of this work culminated in a crystal structure of a truncated PI3 kinase complex in 2007. And this has spurred tremendous drug development efforts in this field. PIK3CA is actually the most frequently mutated oncogene in human cancer. That is a gene whose mutated product causes cancer. And on the right-hand side, this is a histogram showing various cancer types and their frequencies of PIK3CA alterations, mutations shown in green and other alterations shown. And I would like to draw your attention to breast cancer, where PIK3CA mutations are found in about 40% of estrogen receptor, or ER positive, breast tumors. So these are tumors that overexpress the estrogen receptor. Now, what makes these mutations rather complicated to study is that there are a lot of them. They're located all throughout the gene, and they have varying levels of activity. Shown in the pie chart, you can see that the most frequent so-called hotspot mutations, that is, mutations that are very frequent, are found at E542K, E545K, and H1047R. These are the mutation positions. That means that the other mutation part of this pie chart is actually the majority of mutations, about 60%. And as you can see in the lollipop plot in the second panel, these mutations are scattered throughout the gene in all different domains. This is important because this is one way that lipid kinases like PI3 kinases differ from protein kinases, where often in protein kinases, such as receptor tyrosine kinases, mutations are primarily in one domain, the kinase domain. And this makes it complicated to study these PI3 kinase mutations because it necessitates looking at the entire protein complex, not just one domain in isolation. Almost all of these mutations are what we call allosteric. They're not located in the enzyme, in the kinase domain where the ATP molecule, which is the molecule whose phosphate group is transferred to new lipid species, is located. And this, again, makes these mutations rather complicated to study. Finally, as shown in the graph on the bottom, we have some data on activation, the activation capacity of these different mutations as measured by cell growth in a variety of non-transformed or non-cancer cell lines. But we have very little data on drug sensitivity. That is, do these mutations actually predict for drug sensitivity or drug resistance, apart from the most frequent hotspots that are shown in red. I think it's very important to note that we have had many drugs that have targeted this pathway over the decades, starting in, first discovered in the 50s and really explored in the later decades, were so-called PI3K inhibitors. These are inhibitors that hit PI3 kinase, but also other large PI3 kinase-related kinases, such as mTOR, ATM, and ATR. And then this led to more specificity, these inhibitors called dual PI3 kinase mTOR inhibitors, and then finally pan PI3 kinase inhibitors that inhibit all the different isoforms that I mentioned earlier of PI3 kinase. Now, unfortunately, none of these drugs advanced in the drug discovery process due to either less than predicted efficacy, or mild efficacy at the expense of high toxicity. I should note that many other nodes of the PI3 kinase pathway, such as mTOR, has been successfully inhibited and have led to FDA-approved drugs in a variety of cancer types, such as everolimus and temsirolimus, which are FDA-approved in breast cancer, kidney cancer, and pancreatic neuroendocrine tumors. Additionally, we have other PI3 kinase isoforms that can be inhibited, such as PI3 kinase delta, shown on the right-hand side, and this has led to FDA approval of a number of drugs in lymphoma. But PI3 kinase alpha which is the primary driver of breast cancer, inhibitors for these for PI3 kinase alpha were recently developed and have led to an FDA approval of the drug called alpelisib in breast cancer that I'll explain in the next slides. And finally, I put at the bottom a PI3 kinase or PIK3CA mutant selective inhibitor. Many of the toxicities of this drug class are due to on-target inhibition of insulin signaling. And so if we could have a drug that inhibited mutant PIK3CA, as would be found in cancer, but not wild-type PIK3CA, this would be a huge step forward for the field. Here is shown a graph of multiple phase 3 trials, randomized clinical trials that have actually shown a benefit for PI3 kinase inhibitors in PIK3CA mutant estrogen receptor positive breast cancer. And what is rather stymieing is that all of these clinical trials were positive clinical trials. They met their primary endpoint, increasing either pathologic complete response or progression-free survival, with combining a PI3 kinase inhibitor with standard endocrine therapy. But because of these on-target toxicity issues due to inhibition of the insulin signaling pathway, none of these drugs moved forward in the drug discovery process, with the exception of alpelisib, shown in the red box. And this is the clinical trial that led to FDA approval of alpelisib. This was the SOLAR-1 trial, which tested the combination of fulvestrant, a selective estrogen receptor degrader, which is a standard therapy, in combination with alpelisib, a PI3 kinase alpha-specific inhibitor, in patients with PIK3CA mutant metastatic estrogen receptor positive breast cancer. Again, PIK3CA mutations are found in about 40% of patients with ER positive breast cancer, so this is a large patient population. And this clinical trial met its primary endpoint, increasing progression-free survival from about 6 months to 11 months. Now, one important point borne out in this trial as well as prior trials is that PI3 kinase and alpha inhibitors in breast cancer, at least, are not effective in PIK3CA wild-type tumors. And there are other PI3 kinase alpha-specific inhibitors in clinical trials, notably GDC 0077. But this trial was the pivotal phase 3 trial that led to FDA approval of this combination in May of 2019. When I started in the laboratory, I was interested in if there are maybe other factors that may cooperate with PIK3CA mutations to increase response to PI3 kinase inhibition. And this is shown, or this idea is shown in this picture here, where you could imagine that with PIK3CA mutations, that these tumors are sensitive to PI3 kinase inhibitors, as shown in the gray dotted line. But could there be another genotype or another factor that cooperates with PIK3CA mutations to really drive increased sensitivity to these drugs? And the germ for this study came from the identification of an exceptional responder patient to alpelisib by Maurizio Scaltridi's group in 2015. This paper identified a patient who had a long response to alpelisib monotherapy, but then developed acquired resistance. And the nature of this acquired resistance was due to mutations in the cognate enzyme, P10, which antagonizes the effects of PI3 kinase. Now, when I started, I was interested in why this patient may have had such an exceptional response in the first place. And it turned out that this patient was a breast cancer patient who had double PIK3CA mutations in her primary tumor, as well as 14 out of 14 metastases. This graph here shows the variant allele frequency, or the percentage of alleles that contain the mutation that's indicated. And these variant allele frequencies, or VAFs, are graphed one mutation versus the second. And you can see about a 1-to-1 correlation here in both the primary untreated tumor as well as all the metastases. Now, this correlation between double PIK3CA mutations and an improved response was also echoed in a recent phase 1 clinical trial shown in panel B that our group published in Nature Cancer in this year. This was a trial that tested alpelisib in combination with an aromatase inhibitor, another standard endocrine therapy in ER-positive breast cancer. And what we found was two interesting findings. The first was that in a population of 51 patients, 9 patients actually had tumors that contained double PIK3CA mutations. And this was surprising because this phenomenon had not been reported, and this seemed like a high number. Moreover, these 9 patients had an improved progression-free survival of almost a year compared to patients with single PIK3CA mutations about 20 weeks or 5 months. Now, this was not statistically significant due to small numbers, but we thought that this was an interesting signal. We also thought that this signal of improved response may be due to the PI3 kinase inhibitor rather than endocrine therapy alone. Keep in mind, please, that almost all patients receiving these drugs received the combination therapy. We thought that this was due to PI3 kinase because, as you can see in panel C, patients have similar responses to endocrine therapy regardless of their PIK3CA mutational status, whether it's wild type shown in gray, single mutant shown in green, or multiple mutants shown in red, in response to multiple types of endocrine therapy, aromatase inhibitors at the top and selective estrogen receptor degraders like fulvestrant at the bottom. So armed with this data, we asked a very simple question. What is the frequency of double PIK3CA mutations in breast cancer? And what we were shocked to find was that multiple PIK3CA mutations are found in about 10 to 15% of all PIK3CA mutant cancers. This is a series of bar graphs identifying patients in CBioPortal. Most of these tumors are primary tumors. And as you can see, we see about a 10 to 15% rate of multiple PIK3CA mutations in PIK3CA mutant cancers. And this is true in both common cancers like breast cancer and colorectal cancer. But marching down this graph, you can see that even in the rare cancer types, which are shown in the second inset, that we see this persistent signal. And we recapitulated these data also in MSK Impact, which is our internal biorepository that is enriched in metastatic patients. We found in all of our data sets that the vast majority of multiple mutant tumors contain exactly two PIK3CA mutations. Next, we checked to see if these mutations are scattered randomly throughout the gene or if they're enriched at certain amino acid positions. And we performed so-called codon enrichment analysis, asking the question, do mutations at a particular position, are they more frequent in single mutant tumors versus multiple mutant tumors? And as you can see in the pink circles in the left-hand figure, in breast cancer, mutations at position 726, 453, and 1043 are found primarily in multiple mutant tumors compared to single mutant tumors. You can see the common hotspot mutations 542, 545, and 1047 are found just as frequently in single mutant tumors, shown in gray, as in multiple mutant tumors. And what's interesting is that this mutational pattern is different in other cancers, shown in the blue circles. Most of these data here are colorectal and endometrial cancers, where mutations in 88 and 93 in the N-terminus of PIK3CA are more frequent in multiple mutant tumors. And again, we recapitulated these data in MSK Impact in a complementary data set. And so putting these data together, shown in the box, these are the most frequent double PIK3CA mutation combinations that we observe in breast cancer. A combination of what we term a major mutation, 542, 545, or 1047, combined with a second site mutation rarely found alone in breast cancer, 453, 726, and 1043. And we'll come back to these specific combinations a little bit later. Now, we next wanted to find out, are these mutations actually in the same cell in the cancer? And to do this, we performed a clonality analysis using facets, a computational algorithm. And using this algorithm, we found that both mutations are clonal. That is, both mutations are in the same cell. We also performed additional clinical analysis of breast cancers bearing double PIK3CA mutations. And we found that, echoing our initial data, that double PIK3CA mutations are just as frequently found in primary untreated tumors as they are in metastatic tumors. This is an important point, showing that multiple mutations are not simply the manifestation of drug treatment or tumor heterogeneity. And in the bottom bar graph, you can see that double mutations are enriched in so-called hormone receptor positive or estrogen receptor positive HER2 negative breast tumors, and are much rarer events in HER2 positive breast tumors or triple negative breast tumors. Next, we wanted to discover the allelic configuration of double PIK3CA mutations. This is incredibly important because any mutation, any two mutations that is, can be either on the same piece of DNA or on separate pieces of DNA. If they were on separate pieces of DNA, as shown in the middle in green and red and yellow, so-called transmutations, these would code for single proteins with different mutations. But if the mutations were in the same piece of DNA as shown in the red boxes, these pieces of DNA would code for single proteins with multiple mutations. Now, based on our clonality data, we hypothesized that double PIK3CA mutations are in cysts on the same allele. But we were stymied initially when we tried to investigate this because there is no general method to phase mutations far apart in the gene in a single read. And this slide outlines two of the technical hurdles that we had to overcome to answer this question. The first is that most tumor samples are preserved as FFPE, or formalin-fixed paraffin embedded. This procedure degrades DNA into very small fragments comprising about 200 base pairs. Therefore, any mutations that are further apart in the gene than 200 base pairs, which it turns out all of the double mutation combinations that we observed are, the most frequent double mutation combinations that we observed are, that it's impossible to phase these mutations from archival samples. And therefore, to overcome this hurdle, we obtained fresh tumor samples obtained on an IRB, on an Institutional Review Board approved protocol. Secondly, the vast majority of sequencing techniques that we use to determine the allelic configuration of mutations uses NGS, next generation sequencing, or Sanger sequencing, and these are short distance sequencing reads. And we sought a technique that could perform long range sequencing reads to phase these mutations in a single piece of DNA in tumor samples. And so to do this, we turned to a long range sequencing technique. And this is an outline of the technique that we leveraged called SMRT-sequencing, or single molecule real-time sequencing. This is a very exciting tool that has been developed over the past decade by several groups, most notably the groups at PacBio who have pioneered the sequencing technique. And what this technique leverages, shown in panel A, is a highly processed DNA polymerase molecule, which is the engine that sequences DNA. And what this molecule, when engineered with high processivity, is able to do is to sequence circular pieces of DNA and keep whizzing round and round this circular piece of DNA to spit out DNA sequences that can be processed and analyzed to form consensus DNA sequences with extremely high fidelity. This is a very important technical point, that this sequences amplicons of circular DNA, because the vast majority of DNA that we send for sequencing in the laboratory is linear pieces of DNA. And of course, a DNA polymerase molecule can sequence that linear piece of DNA, but then it will fall off. And what this sequencing technique allows for is multiple tens of thousands of reads of circular pieces of DNA. And this technique, shown in panel B, can sequence up to 30 kilobases of DNA, an extremely long length of DNA, in a single read. And this is very exciting technology because, as shown in the previous slide, every single one of our double mutant combinations was very far apart in the gene. And so we thought this could be a technique that could actually help us figure out if these mutations are in the same allele or not. Now, working with our friends at Mount Sinai School of Medicine and Bobby Sebra, we devised a protocol where we initially identified metastatic patients who had double PIK3CA mutations using a next-generation sequencing platform, MSK Impact. We consented them to our biospecimen protocol, and these patients graciously underwent fresh metastatic tissue biopsy. And we performed the workflow shown here. We first optimized this workflow to determine the allelic configuration of several parental cancer cell lines that have multiple PIK3CA mutations. And in data that I'm not showing you here, we determined that many of these cell lines have mutations in cis on the same allele, and some of them actually have mutations in trans. But we were able to use this workflow to optimize this technique. After optimizing this technique, we turned to our patient samples to determine the allelic configuration of the double mutations. And this is actually our data of each of 6 patients bearing 6 different double PIK3CA mutation combinations. And the bottom line is that all of these mutations are in cis on the same allele. I'd like to walk you through a little bit of this data. In the first row, we have a patient with E545K, E726K. This is the most frequent double mutation combination that we see in breast cancer. And looking at the colored bars on the right, you can see that 58.1% of amplicons contain mutations in cis on the same allele, which are shown in the red squares. Now, 39.5% of the amplicons have wild-type DNA. This is to be expected because all tumor biopsies are not 100% pure, so we're going to have some wild-type DNA in there. But only 2% amplicons contained E726K alone, shown in yellow, and only 0.2% in this sample contained E545K mutation in trans. And if we march down to these other patient samples, you can see that the vast majority of the mutant amplicons are in cis on the same allele. We also used orthogonal techniques such as colony sequencing, next-generation sequencing, and RNA sequencing to validate rare double mutations that are actually very close together in the gene using these short-read techniques. And to our knowledge, this is the first use of SMRT-sequencing to phase multiple alterations in solid tumors. And therefore, all of these data together show that, at least in breast cancer, the vast majority of double PIK3CA mutations are in cis on the same allele. Now, armed with this data that these mutations were in cis, we proceeded to functionally characterize the effects of double mutations. We focused in panel A on the mutation combinations shown in the second gray box. And in all of these functional assays, we tested the wild-type gene, mutation A, mutation B, and mutation A plus B. And so in panel B, we assembled a series of isogenic cell lines. These are cell lines with, that only differ in their PIK3CA mutation and otherwise are genetically similar. And we performed Western blotting looking at cell signaling pathways. And as you can see in the Western blots, double mutations, as I'm showing here, double mutations have increased signaling in the PI3 kinase pathway, both proximally at the level of phospho-AKT and distally at the level of phospho-S6. What's pretty striking is that at the level of phospho-S6, you can see that the Western blotting, that these bands are only present in the double mutants and are really not present in the wild type or in single mutants. And so therefore, double mutations increase PI3 kinase pathway signaling compared to single mutations. We also had similar findings in different cell lines. Here we tested MCF10A cells. These are normal epithelial breast cells. But we also had similar findings using NIH3T3 cells, which are mouse fibroblasts, as well as MCF7 ER-positive breast cancer cells. We looked at signaling kinetics, that is signaling over different time points, and we found that at a 48-hour time point, the double mutants have increased signaling compared to the single mutants and wild type, as shown at in this series of Western blots. We next tested to see if this increase in signaling correlates with an increase in cell growth, and we see that yes, that this is true. You can see that the double mutants have increased cell growth in panel A by crystal violet staining, and this is quantified in panel B. And here we're starting to see synergistic effects where the effects of the double mutant are much greater than the additive effects of each constituent single mutant alone. We also tested if these mutations expressed in trans, that is on separate alleles, what the effects of these are. And we found that in trans, these double mutations have similar signaling levels to the single mutants, as shown in the left-hand panel, and do not have increased cell signaling, or excuse me, increased cell growth in the right-hand panel compared to the mutations in cis. We looked at the effects of our mutations in mouse models by creating allograft that is taking our cell lines and injecting them into mice and monitoring for tumor growth. And as you can see, the double mutant shown in light blue, E726K, H1047R, this confers increased tumor growth compared to single mutants and wild type in a mouse model, and also leads to increased cell signaling, as shown in Western blotting in panel B, and by immunohistochemistry in panel C. Next, we wanted to investigate the mechanism of how the PI3 kinase complex is modified by these double mutations. And so to do this, I'd like to first take a big step back to investigate or to discuss what's known about how these two different proteins, P110 and P85, function. And work from Jonathan Backer's group, a very elegant work, has shown that P110, which is a catalytic subunit, again coded by PI3K, is both stabilized and inhibited by P85, the regulatory subunit. And this is shown in panel A. This is pulse chase, showing that P110 by itself degrades at about 2 hours, while in the presence of P85, this complex is stable for at least 5 hours. And this increase in stability correlates with a decrease in activity, as shown in panel B. And this is quantified in panel C, where P110 by itself has high catalytic activity. In the dotted line, when bound to P85, this catalytic activity is decreased and is partially rescued with the addition of a phosphopeptide. Now, the addition of a phosphopeptide is a mimic of what happens normally in a cell, where a receptor tyrosine kinase, such as the insulin receptor, is phosphorylated and then binds to PI3 kinase for downstream signaling. And this phosphopeptide binding also leads to increased lipid binding. Now, this graph, this is just a pictorial representation of the data that I showed you in the prior panel, that in the presence of P85, the wild-type PI3 kinase complex has low levels of kinase activity, and that sequentially, with the addition of phosphopeptide, this opens up the protein kinase complex, leading to increased lipid binding. And when P110 is by itself, that is as a monomer, it has very high catalytic activity. So now, amidst this framework, how do the single hotspot mutations E545K and H1047R function? And this is a pictorial representation of a number of seminal, beautiful biochemical and biophysical experiments done by many groups over the years. And what these groups have shown, notably Mario Ansel's group and Roger Williams's group, is that E545K functions as what we term a disruptor. It disrupts the inhibitory interactions between P85 and P110, opening up the protein complex. And other mutations that are at the P110-P85 binding interface have a similar mechanism of action. In contrast, H1047R, which is located in the C terminus at the end of the P110 protein, functions as what we call a binder. It increases lipid membrane binding, and this is pictorially represented here. And other mutations that are in the C terminus of P110 have a similar mechanism of action. So therefore, this has established the most frequent hotspot mutation mechanism. And now, what is the mechanism of these other second spot mutations that we identified? In panel A, this is a domain schematic showing the location in green of the second site mutations, 453, 726, and 1043. In panel B, this is the crystal structure with all of these three different mutational positions and 545, 1047 shown as black spheres. As you can see, 453, E545 is located at the binding interface between P110 in blue and P85 in orange. It's right between the orange and the blue. Same thing with 545. 1043 is located very close to 1047 in the C terminus of PI3K. And 726 is located in this loop here, far away from these other mutation sites. Therefore, we speculated that E453Q functions as what we term a disruptor, just like E545K. And M1043L functions as what we term a binder, like H1047R. However, E726K, nothing was known about what this mutation might be doing. But based on the crystal structure shown in panel B, as well as in this slide showing the alignment of multiple crystal structures, you can see that E726 is located in this loop that is right by the putative lipid membrane plane. Now, E726 is an amino acid called glutamic acid, which is acidic and is negatively charged. If mutated to a K, which is a lysine residue, lysine is a positively charged amino acid. Now, this is important because lipid membrane phospholipids are negatively charged phospholipids. And so we would predict that if we have a mutation to a positively charged amino acid, this may increase affinity with a negatively charged phospholipid in a membrane. Therefore, we speculated that E726 would function as a binder. And together, these classical papers in the field, combined with our investigation of the crystal structure vis-a-vis the mutations that we have identified in our study, led us to the hypothesis that double mutants activate PI3 kinase by both increasing P85 disinhibition, that is, increasing the disruption, as well as increasing membrane binding. Now, to investigate this hypothesis, we prepared recombinant full-length human PI3 kinase mutant protein complexes and subjected them to a variety of biochemical experiments. To investigate P85 disinhibition, we performed an assay called thermal shift. This is an assay that measures protein destabilization, and we interpret that as a proxy for P85 disinhibition. This is the Western blotting. What we do is we take our proteins and heat them on a temperature gradient from 46 degrees to 63 degrees. And the protein complexes are blotted by Western blotting to look for the temperature at which the protein denatures. You can see that wild type, we see 5 spots on our temperature gradient, but only 4 spots with E545K, showing that E545K denatures at a lower temperature than wild type. And this is the raw data of our experiments. And as you can see, the double mutants, which is shown in the lowest row in each panel, the double mutants denature at a lower temperature than the single mutants or wild type. This is quantified here in panel A, where you can see that the double mutants have lower melting temperatures than the single mutants or wild type. In panel B, we performed lipid kinase assays using radioactivity, directly measuring the product of the kinase reaction. And we showed that the double mutants had increased lipid kinase activity compared to the single mutant. And finally, we looked at lipid binding by preparing liposomes, that is circular vesicles of lipids with different lipid species, and we measured their binding with our recombinant PI3 kinase protein complexes. And as you can see in panel C, in the Western blots, which is quantified in panel D, all the double mutants had increased lipid binding compared to the single mutants or wild type. Therefore, putting all of these data together, these biochemical data suggest that double PIK3CA mutations increase kinase activity through a disruptor-binder combination mechanism. Next, we wanted to investigate in our cellular models how double PIK3CA mutations may modify sensitivity to PI3 kinase inhibitors. We took our cell lines and exposed them to alpelisib. The Western blots I'm showing you here are a short-term 1-hour drug treatment. And as you can see, in the absence of drug, as shown in the minus columns, the double mutants have increased PI3 kinase signaling compared to single mutants. However, in the presence of drug, as shown in the plus columns, the signaling is abrogated under all conditions. And we showed similar findings in our other cellular models. Now, this was a short-term drug treatment for 1 hour. However, when we expose these cells to much longer durations of drug treatment, in this experiment, 4 days, you can see that the double mutants are actually more sensitive to PI3 kinase inhibitors than single mutants. For clarity, I've broken this up into the double mutants containing E545K in the top panel and the double mutants containing H1047R in the bottom panel. In the top panel, you can see that the double mutants E545K, E726K, and E545K, M1043L, as shown in the purple and brown curves, are more sensitive. That is, the cells are killed with a lower concentration of drug compared to E545K, and in turn, E545K is more sensitive than these rare single mutants found rarely by themselves in breast cancer. And this is true in the bottom with the H1047R containing double mutants. And these patterns of drug sensitivity by Western blotting and by cell viability assays were recapitulated, showing almost identical results using a structurally dissimilar PI3 kinase inhibitor, GDC 0077, which is now in late-stage clinical trials. We also tested our transmutant cells and showed that the transmutants are not, in fact, more sensitive to alpelisib versus single mutants. In these curves, the transmutant is shown in the light blue dotted line, and you can see that the drug sensitivity of the transmutant is very similar to the single mutant. Finally, we wanted to validate our hypothesis connecting and correlating the increased response of double PIK3CA mutant tumors to PI3 kinase inhibitors in a clinical setting. And to do this, we teamed up with Genentech to perform a secondary analysis of the Sandpiper clinical trial. Now, this clinical trial was similar to the SOLAR-1 trial regarding patient inclusion criteria. It had a very similar patient population. And this trial tested the addition of tacelicib, a PI3 kinase alpha inhibitor, to fulvestrant, which is an ER degrader, in PIK3CA mutant metastatic estrogen receptor positive breast cancer. This trial enrolled 631 patients, and 339 patients had PIK3CA mutations. Of those 339 patients shown in gray, 66 patients shown in blue had multiple PIK3CA mutations, while 273 patients shown in orange had one PIK3CA mutation. I'd like to point out that for this analysis, we used circulating tumor DNA to determine PIK3CA mutational status. And we did this because many patients with ER-positive breast cancer have bone-only metastases, and bone biopsies are very difficult to analyze due to the need for decalcification. And so therefore, we use ctDNA, which we rationalized would allow us to analyze a larger number of patients. This is a so-called waterfall plot of the patients on the tacelicib arm of the Sandpiper trial. Each bar represents a single patient, and positive y-axis, shown in the left-hand side, correlates with corresponds with tumor growth, whereas negative y-axis corresponds with a tumor shrinkage. Now, first, ignoring the colors, we can see that more patients had tumor shrinkage than tumor growth. This is consistent with the fact that this was a positive clinical trial. Again, this drug did not move forward in the drug discovery process due to increased toxicity. And then looking at the colors, we can see that multiple mutant patients shown in blue have more of increased tumor shrinkage compared to single mutant patients shown in orange. And this is quantified in these bar graphs. In panel A, this is the total mutant population, or the total population of patients with PIK3CA mutations. You can see an increased response rate with the addition of tacelicib, and this is the whole patient population. And when we break it up into patients with single PIK3CA mutant tumors versus multiple PIK3CA mutant tumors, we see that the patients with single PIK3CA mutant tumors had an increased response rate, but this was not statistically significant, while the patients with multiple PIK3CA mutant tumors, shown in blue, had an increased response rate, which was statistically significant. Now, I'd like to point out that, of course, this was not the primary endpoint of this Sandpiper clinical trial to investigate the response of double PIK3CA mutant patients, but this reflects the best available clinical data at this time to ascertain and elucidate our hypothesis. And we await publication of the Sandpiper clinical trial to determine the effect of double PIK3CA mutations on progression-free survival in this population. And so together, these data suggest what we term a mutational dosage model for PI3 kinase activation and inhibition in breast cancer. Wild-type PI3 kinase alpha, as shown in the third row of this box, leads to increased AKT signaling, while mutant PI3 kinase leads to even more increased AKT signaling, and double mutant PI3 kinase leads to even more AKT signaling and increased cell growth. And this is important because it correlates with a clinically meaningful phenotype of increased response to PI3 kinase alpha inhibitors for double PI3 kinase mutant tumors. And these data point to a second mechanism for oncogene activation of double PIK3CA mutations in cysts. And so in conclusion, I've shown you that double PIK3CA mutations are a novel and frequent genomic alteration in PIK3CA mutant cancers. They're in cis on the same allele. They activate PI3 kinase signaling in growth more than single mutants. We've shown this in a variety of models, and they do this through a novel combination disruptor-binder biochemical mechanisms. We've shown that cis mutants increase response to PI3 kinase alpha inhibitors compared to single mutants in breast cancer. And we believe that this is a rationale to test PI3 kinase alpha inhibitors in multiple PIK3CA mutant cancers. With this, I would like to acknowledge many people, Jose Baselga and Mauricio Scaltriti, for providing an exceptional laboratory environment to investigate this question, Lou Cantley's lab at Weill Cornell for tremendous help with our biochemistry experiments, my fellow breast oncologists on the Memorial Sloan Kettering Cancer Center Breast Medicine Service for helping me obtain patient samples, my many other collaborators at MSKCC, Bobby Sebred, Melissa Smith at Mount Sinai Genome Sciences. They were tremendously helpful and critical for helping us with the single molecule real-time sequencing, or SMRT-seq. My other collaborators, Raul Rabadon and Maura Dickler, Lori Friedman, previously at Genentech, and the entire group at Genentech for collaborating with us on the Sandpiper clinical trial analysis. And finally, to the many patients all over the world who have obtained biopsies with us and have enrolled on PI3 kinase inhibitor trials over the years. This is a list of my funding sources, and at this time, I would be happy to take any questions. Thank you very much.